Okay, my entire setup has changed, so hopefully this will work. Just to set the stage for you, I'm in the basement of my university with the lights turned off, looking at a projector with a gaming headset on that I quote borrowed from a friend. So I hope that this will work a little bit better and it won't be as glitchy. Bear with me, but we're working on it. Okay, so we were talking about opioid neurotransmitters. We had mentioned that they tend to be antidepressants. I also wanted to mention that they mediate complex behaviors. Right? Well, complex behavior is pretty vague, so a couple examples of this. Uh, One example might be sexual arousal. Another example of this could be submissive and dominant relationships. So these types of behaviors. Okay, typically opioid neurotransmitters are involved here, but probably where they're known best is for their ability to stop pain. So analgesia is probably the biggest one, right? Which is essentially inhibition of pain. You may have heard of this, right? Like if you take a topical analgesic, that's something that you put on your skin to eliminate pain. Now, um, like all other opioids, or excuse me, like all other peptides, opioids work through G protein coupled receptors. Right, so all of these all work through GPCRs. Now, there's a particular subcategory that opioids bind to. There are three classes of receptors, right? Three classes of G protein coupled receptors. Right, but the main one that I think we'll talk about is what's called the mu opioid receptor, right? The Greek letter mu. Oops. Okay, and the mu opioid receptor is important because it's the target for drug reward mediated by the opiates. So I think, right, that opiates can be highly addictive, right? They're great for this, they're great for analgesia, but they're addictive. And the reason is because, right, through this mu opioid receptor, there's reward circuitry in the brain that gets strengthened every time these receptors are activated by these opioids, okay? So that reward circuitry is mediated by opiates and it works through this mu opioid receptor. It's a G protein coupled receptor, okay? Is there anything else we want to say about opioids? Well, I guess we could mention in general, like all other peptides, right? They're really, really potent. And so what do I mean by this? Typically, they can work at very low concentrations that they require only nanomolar to low micromolar, excuse me. So for example, a small molecule neurotransmitter may be required at, oh, I don't know, something like 500 micromolar, whereas these opioids may only be 50 nanomolar, right? So you're talking 1,000 or even 10,000 fold difference in concentrations, and yet can still have the same sort of potent effects. All right. So really, this is it for what we think of as traditional or classical neurotransmitters, right? Things that are released based on calcium entry, things that are packaged in vesicles. So we'll talk about a few different types of neurotransmitters now that aren't that way, that are unconventional. All right. And so the first one, oops, I have to erase. bear with me here. The first one that we're going to talk about, or the first category, are endocannabinoids. Let's see. Okay. So endocannabinoids, you see this word endo, right? So you would be thinking right here, endogenous, and that's exactly what these are. These are endogenous signals. Right, so they're coming from within the body, and they activate or bind to particular cannabinoid receptors, right? So this is the receptor. Receptor, okay? Now, these receptors are also targets for a compound called delta 9 tetrahydrocannabinol. Yeah, right. You're thinking, well, why would you need to know that? Well, that's because you have THC, right? You might have heard of THC. Oops, TCH. THC, right? This is the compound that's active in cannabis, right? So this is the psychoactive. That's in the cannabis plants. So this is where a lot of research on this receptor has been focused now. But we'll talk about it with respect to the endogenous compounds that activate it, right? And so these endogenous compounds are produced by the enzymatic degradation of lipids. So we're not going to talk a lot about this pathway, right, the enzyme that's involved or anything, but know that it's an enzymatic cleavage. I butcher this, enzymatic degradation of lipids. So the fact that it's a lipid byproduct is important because that means that it can freely cross cell membranes. It doesn't require a surface receptor, right? And so everything else, a peptide's not going to cross cell membranes, but a lipid, of course, a lipid can. And so although the release mechanism is unclear, we think that it moves via diffusion to its receptor postsynaptically. Okay. Once that signal is done, right, that it can be degraded by an enzyme that's within the postsynaptic neuron. So that's unique, right? Typically we've seen presynaptic or glial degradation. This degradation is postsynaptic. Right? And the idea here is that once it can cross freely into the postsynaptic membrane, and there's an enzyme here called FAAH, fatty acid hydrolase. And it's the fatty acid hydrolase, right, FAAH, that's going to break this down. So let's spend a little time talking about the receptors for endocannabinoids. There are two. Try to get this right here. Whoa. So the two receptors are CB1 and CB2. Let's see here. Uh-oh. Timeout. Okay, so receptors. These are G-protein coupled receptors. We're going to talk a lot in Chapter 7 about G-protein coupled receptors. So I know that I've stated this a whole bunch of times, right, that it binds to a receptor and on the cell's membrane and that activates a G-protein. We're going to talk a lot about the downstream targets. I haven't been very specific about that, but we'll get to that stuff. Okay. So in any case, right, this G-protein coupled receptor, the majority of what we know about endocannabinoids and how they work through receptors is through the CB1 receptor. We think that that's more physiologically important, or at least it's much more common. Okay. And the receptor structure is pretty similar to that of glutamate or acetylcholine. So the structure is like glutamate receptors that are G protein coupled, acetylcholine receptors that are G protein coupled, and then they've got 7 transmembrane domains, right, that intracellularly they associate with the G protein, extracellularly they bind the ligand. Okay, now this is where synthetic agonists and antagonists have proven very useful in study, right? There are multiple synthetics that, again, people may take for psychoactive effects, but they're useful in the laboratory setting as well. What they do is sort of interesting. They serve as retrograde messengers and influence GABA release. So functionally, right, is that they're retrograde. So if you think retro, maybe you think backwards, and that's exactly how these work, right? They work backwards to influence the presynaptic cell. Retrograde messengers, right? And what they do is, and I'll show you how they do this in a second, that they influence GABAergic cells. Right, so there's what we think is maybe a co-release mechanism that potentially endocannabinoids are released at the same time or near the same time as GABA release. Neurons. It's neurons, sorry. So here's what we think happens, right? We think that a depolarization typically causes increase in a postsynaptic cell. Right, so in a postsynaptic cell, right, as this cell is depolarized, calcium enters. Postsynaptically. Okay, so this is what I mean. So if we're looking at A right here, right, if we stimulate this inhibitory interneuron, right, it's inhibitory, so we think it would release GABA, and it does. And what that GABA leads to, and we won't get into the mechanism, is an increase in this cell's calcium. So calcium, right, we think enters into this postsynaptic cell upon GABA release here. Okay. Now, one of the things that was required in the synthesis pathway for endocannabinoids is calcium. So I mentioned that there's that enzyme that's important in cleaving lipids. That enzyme is calcium dependent, right? So this is able to activate the enzyme right postsynaptically, which allows for endocannabinoid synthesis. So we'll just say EC synthesis. Okay. What happens next? Well, now that endocannabinoids are made, right, they can be released because they can permeate through and they feed back. So the endocannabinoids, right, they're not released through a vesicle, they feed back to work on this presynaptic neuron. Okay, and they bind presynaptic receptors, right? Retrograde, they go backwards and work presynaptically. And this reduces, right, the amount of GABA that's released. So there's a decrease in GABA. Maybe we'll put that here too, right? So the effect of on the presynaptic cell is reduced GABA. All right, and we'll start the next little video here looking at some data that shows exactly how this happens.